Hello everyone, and welcome back to AP Bio with AP Playground. Today we're covering topic 3.1, enzymes. Today is all about learning what enzymes are and how they work, specifically focusing on explaining how enzymes affect the rate of biological reactions. Our suggested skills are concept explanation 1B, explain biological concepts and processes, and we also have questions and methods 3C, identify experimental procedures that align with the question, including dependent and independent variables, appropriate controls, and justifying controls, which is a question that will often be asked on the exam where you have to determine within an experiment what is the dependent variable, what is the independent variable, and what changes could be made to the experiment to make it better, and what justifies those changes. What is an enzyme? An enzyme is a biological catalyst. And a catalyst is a substance that can change the rate of a reaction, and it isn't used up in the process, so it can be used repeatedly. So enzymes aren't one and done products. They can be used over and over again, and they're going to speed up the reaction. And most enzymes are proteins, which means they follow properties of proteins such as our primary, secondary, tertiary, and quaternary structure, and they're also susceptible to denaturation, which we'll talk about in a second. Some enzymes are ribozymes, which are enzymes that are RNA, so they are nucleic acids and not proteins, but the majority are proteins. And enzymes lower the activation energy of a reaction, which is the amount of energy it takes to start a reaction. If we look at this reaction here, you can see that in order for the reaction to begin occurring, some energy is required to start it, and this is the activation energy. However, with an enzyme, the amount of activation energy needed to start the reaction is going to be much less, and the product ends up being the exact same. So, how do enzymes work? Enzymes act upon substrates. Those are going to be the inputs to the reaction. And enzymes can combine substrates. They can separate substrates and they can change substrates. So they speed up the reaction like we talked about. And the part of the enzyme that binds to the substrate is known as the active site. And this is going to form the enzyme-substrate complex, which we can see here. Substrate is going to attach to the active site of the enzyme, and that's going to form the enzyme substrate complex, and we're going to have products as a result. And of course, the enzyme is not used in the reaction. It can be reused. But another key thing to note is that the active site is specifically shaped for a certain substrate. So there's high specificity to what substrate and what enzyme can work together. Like stated previously, each enzyme has specific substrate or substrates plural it can form an enzyme substrate complex with. And so, discussing further on how an enzyme works, we look at the induced fit model. The substrate and active site have matching shapes and attracting charges, like we talked about, and as the substrate is attracted into the active site, the enzyme will make a conformational change to make them entirely compatible. So, it's not a direct fit, but what will happen as the substrate enters the enzyme will make a conformational change, which will make the substrate and enzyme shape and charge entirely compatible. And so when we're talking about a separation reaction, the enzyme is able to put pressure on the substrate and help break those bonds easier. When we're looking at a combination, the enzyme is able to bring the two substrates closer together and help the formation of bonds. And this is how it lowers the activation energy. Now, having a list of properties of enzymes, we talked about. Enzymes largely speed up the process of reactions by lowering the amount of activation energy needed to start the reaction. Enzymes are specific. We discussed this in the induced fit model. Only certain substrates can bind to specific enzyme active sites. Now, the catalytic reversions are reversible, meaning that if an enzyme is going to break apart a substrate into two products, there's usually going to be another enzyme in the cell that will then put those two products together to make the original input. And this is a regulation ability because it helps in case there are too many or too little of a certain compound within the cell. And lastly, like we talked about, enzymes follow protein properties, so they can be denatured. Lastly, we should take a little look at the types of reactions that enzymes can assist with by lowering the activation energy. First of all, we have a catabolic reaction. And a catabolic reaction is where we're breaking bonds. We're breaking a molecule, and this is going to release energy as bonds are broken. This would also be known as an exergonic reaction or exothermic, which releases heat. And the conformational change of the enzyme is going to place stress upon the bond, lowering the activation energy needed to actually break the bond. An anabolic reaction, on the other hand, is making a molecule. It's forming a bond. It's going to consume and store energy within the bond as the bonds are formed. And this is an endergonic reaction or endothermic. It's storing heat. And the conformational change forces the two substrates together, again lowering the activation energy of the bond formation. And the overall change in energy, whether or not enzymes are used, is the same. So if we are performing a catabolic reaction and the bonds are broken, with or without an enzyme, the amount of energy you get from breaking those bonds is the same. Same thing with an anabolic reaction. The amount of energy you need to store the energy within the bond is going to be the same. However, enzymes make this occur faster because of their ability to lower the activation energy. So there you have it. You should know now what an enzyme is, their properties, and the induced fit model in creating conformational changes to lower the activation energy.